Hi everyone, I think I'm online. So there was some issues going on. Um, am I online? Yep. Can you guys see me? Anyone? Anyone is there? Yeah. So I'm online right now. Sorry if it is a sorry if it is a big problem. I mean, sorry if it is a okay. So once I'm online, I don't know how many of you guys are here right now, but let's start. You can always look at the recorded lecture. So today I was thinking of talking about immunology. So in this span of two courses, we are going to finish all the immunology concepts. So right now, we shall start with cells and the organs of the immune system. We would also look at all the questions that are important for IIT JAM, the topics that are important for IIT JAM. So whenever you're having a time to look at the repeat podcast, you can look at it. So without wasting further time, we can begin. So right now we're going to talk about organs of the immune system, right? So if we talk about the organs of the immune system, this is super important because without knowing this, it is impossible for me to explain other stuff, right? So let's first learn about it and see which part is important, and then we go to the most relevant part, which is important for IIT JAM. Okay? So here, the organs of the lymphoid, the organs of the immune system can be compared to the defense system of our body, right? And we are going to always look at immune system from a perspective of a defense system. In that context, our skin is the first physical barrier. That means the skin is just like a fence which is present in the border of our country. So what our skin does, our skin protects us from many pathogens, many bacteria and harmful agents by several proteins, peptides, by secreting several proteins and peptides like lysozymes, like calprotectins, defensins, etc. So this is fascinating that our skin has capabilities to get rid of the microorganism, but there are also some commensal microorganisms which are present in our skin. But most of them cannot grow because our skin has a very low pH and all of these proteins with them inside them, right? So that's why our skin is not a good place uh, where microbes can grow. Moving on forward, all of these proteins, you don't need to remember the name of it or nothing of it, but try to understand the way skin works to protect our body is by secreting these kind of peptides. These are called antimicrobial peptides like lactoferrin, defensin, calprotectin, all of these things, okay, that generally harms the bacteria and kills the bacteria. Now we talk about two important terminologies which is really important to know. First of all, we're going to talk about the primary lymphoid organ, and second of all, we are going to talk about the secondary lymphoid organ. Because in order to understand the immunology, we need to begin from the basics. For primary lymphoid organ, if what comes in your mind when I talk about primary lymphoid organ, that means that primary lymphoid organs are the places where immune cells are born and they can at least spend some time of their developmental phase there, and later on maturation takes place in the secondary lymphoid organ or in tertiary ones. So let's look at what primary lymphoid organ we have in our body. So the prevalent primary lymphoid organ is the bone marrow. Especially bone marrow is present in case of the big bones such as your humerus, your radius ulna, etc. So inside the bone marrow, if we cut a cross section, what we are going to see is it has blood vessels running through the bone and also bone cells known as endosteum cells, and also it has a niche of all these bone cells known as endosteal niche. Niche means or a locality, or let's say an address. Now you have vascular niche around the blood vessels. I'm telling you guys because this is important. Bone marrow contains several hematopoietic pluripotent stem cell. Now this hematopoietic pluripotent stem cell give rise to all of our immune system. So everything is born from the hematopoietic pluripotent stem cell. As we go further in our lecture, we would know that what hematopoietic pluripotent stem cell is capable of doing. Okay, so hematopoietic pluripotent stem cell give rise to two lymphoid progenitor on one side and myeloid progenitor in other hand side. Now from lymphoid progenitor, your adaptive immune system components such as B cell, T cell, natural killer cell, etc. develops. Whereas from the myeloid lineage, your blood cells such as RBCs, WBCs, in terms of WBCs, your basophils, neutrophils, and mast cells develop from the lymphoid progenitor. And most of these cells are actually the cells of your innate immune system. So broadly, we understand hematopoietic pluripotent stem cells give rise to two different kind of progenitors, which ultimately give rise to several components of the immune system. Okay, so next we talk about thymus. So thymus can be understood as the training school of the T cell. It's also a primary lymphoid organ, okay, because a chunk of the developmental phase of the T cell takes place inside the thymus. And we would learn in this lecture that how thymus is important for T cell development. So that is included in today's course. So let's talk about it. So thymus is a training school of the T cell. There they learn very important things such as how to recognize antigens, which is displayed on MHC molecules. That's important training. If they don't learn this training, they cannot do it when they really encounter pathogens. So as if a particular military person doesn't know that how to fire a gun, he cannot do it in the battlefield itself. So they need a training how to fire a gun and how to shoot, right? It is very similar for the T cells, and the training is given by the thymic epithelial cells, which are the instructors of these T cells. As we go along, we would learn more about it. So for now, it's just a quick overview. Next, we talk about, like, there are several regions inside the thymus which have different cell compositions. These are known as cortical epithelial cells or medullary epithelial cells of the thymus. So they give important lessons to these naive T cells, or not naive, I would say very mature state of T cells. And as a result, they would help them to become mature and help them to gain the capability of determining a particular antigen displayed on MHC molecules. Now in thymic, there are several selection procedure going on which we would learn later, but at the end of the day, what happens in the thymus is like T cell either becomes a cytotoxic T cell, which is CD8 positive, it has the CD8 co-receptor, or it becomes the CD4 positive helper T cell, which has the CD4 receptor. Now, I mean, CD4 and CD8 both have their different function. For example, CD4 is the helper one, so it basically works hand on hand with the B cells, and that is how they work. Okay, so in the secondary lymphoid organ, as we are going to talk right now, the secondary lymphoid organ are the organ which help the immune cells to grow, right? For example, here we are giving the example of lymph node. Lymph node is spread pretty much all across your body. Now what happens is the lymph node is giving you a way, a particular, it's like an army base, okay, or army base camp, you might say, because inside the lymph node our T cell, B cell, which are the warriors of the immune system, they reside. Now inside the base you would have several barracks, right? Some for land troopers, some for air forces, etc. So exactly inside the thymus, inside the lymph node, you have several regions such as follicular areas where B cell reside, and paracortical area where T cell resides. So T cells and B cells reside in specific regions, okay? Just like you have army barracks, they are also different type of warriors, so they have different space to stay. So they are spatially segregated. And in this lymphoid follicles or in this paracortical area, what happens, dendritic cell can come in and they can interact with these cells. Next we talk about the biggest secondary lymphoid organ of our body, which is spleen. This is an important concept in JAM point of view, because they can always ask you which is the biggest lymphoid organ in your body, which is spleen. Now spleen is an organ which is just under the liver, and it is widely known for eliminating bloodborne pathogen, and it's a graveyard for RBCs, right? Apart from these, spleen also has regions for T and B cell, for their residence. So spleen is just compared to a very big army base having a lot of barracks and all. Okay, so inside the spleen you have white pulp. So these white pulp, which is also known as like the periarteriolar lymphoid sheath, which is a actually a residence for the T cell. So they sometimes they might ask that inside the spleen, which is the residence of the T cell. Now, the closely associated with these area, there is a B cell rich lymphoid follicles also present in the spleen, just like we have seen in the inside the lymph node. Here you also have same sort of follicles which are forming the germinal centers. But what you really need to remember is spleen is the biggest lymphoid organ in our body, and which really help the B cell and T cell to reside there. It's a big army base, army headquarters, you might say. Other than that, there are other small peripheral lymphoid organs such as mucus associated lymphoid tissues or gut associated lymphoid tissues, which are present just under the gut in the intestine. One such example is Pierre's patch. They might ask this particular information from you. Now then we move on to understand cells of the immune system, because we have pretty much talked about the organs of the immune system, and these are pretty much the catalog of what type of cells you find in our immune system and how we can compare them with the defense system of our country, right? So here we have our B cells, T cells. These are actually like special operative forces which really go down in the field and do all the heavy job of killing. And today's lecture we are going to talk about all of these. I'm assuming that you guys have no idea about immunology, and then I'm going to build up from really basics, such that when we go into the advanced concepts you don't feel left out. So then other than that, the other cells which were derived from the myeloid lineages such as basophil, neutrophil, mast cells and eosinophils, all of these things plays several important role in the immune system. As we go along we would see. So first let's talk about, like, let's try to get the similarity between defense system and the immune system. So here bone marrow is the, as I already just reminding you that bone marrow is the region where all these development of the immune system takes place, right? So inside the bone marrow there are progenitors which give rise to these things. Just reminding you, we have already discussed these things. So now let's talk about monocytes. I mean, you know that monocytes are actually derived from myeloid progenitor cells. Now monocytes are formed inside the bone marrow, which are known as like band monocytes or immature monocytes. Later on the monocytes are secreted in the bloodstream, then they undergo a little bit maturation process. But see that image below, where the monocytes is in the blood vessel, they have a complete different morphology. But when they are in the tissue space, they are known as macrophages. And they have total different morphologies, having a lot more vesicles, mitochondria, and a bigger nucleus. Now, macrophages are just like a patrolling police officer. Why I call them a patrolling police officer? Because underneath your skin, they are moving all across. Not only macrophage, there is another division of these patrolling police officer, which we call dendritic cell. But let's say a particular nail is pricking in your skin, and with that rusty nail, a lot of pathogens entered your body. then macrophage is the first one to see that there are invasion, and they try to kill that pathogen by several way. And not only they kill, they also call for backup. The way pathogens are killed by macrophages is called oxidative burst. Generally, macrophage generate reactive oxygen and reactive nitrogen species inside them, which would help to kill the pathogen. So we understand the patrolling police officer is the macrophage. It kills the pathogen by which, but honestly, they don't have immense power to kill them and neutralize all of those pathogens. So they also know that. And that's why they always call for backup. What they do is they secrete cytokines for call for backup. So they are pretty much similar to a police officer who are not heavily armed, but can to some extent encounter some invasion, right? But here we are going to talk right now about CD8 cytotoxic T cells. CD8 positive cytotoxic T cells are the subclass of the T cells, which are also known as the special operative SWATs of the immune system, because they are like killers. So their job is to kill. And in this video, it would be more evident that how they kill actually. And I'm not getting into the details of it, but in my lectures, there are many of these things which you can take a screenshot and use like a flashcard. So this quickly shows the how what is the mechanism by which CD8 positive T cells kill. Okay, coming to the CD4 positive T cells, they are also army, but they are somewhat supportive forces. What they do is actually interact with the B cells. Now B cells are the land troopers. They get into the action and they have their B cell receptor, which is none other than the antibodies, membrane bound antibodies, membrane bound IgM, and that is how B cells can ultimately mature into plasma cells, which is an antibody secreting structure. Now B cell development takes place in the bone marrow. Later on, B cells move to the lymph node where they learn many of the things and undergoes many changes. I'm just building a platform such that I can tell you about all of these changes as we go along. Now T cells and B cells have different zone inside the lymph node for their residence, right? The lymphoid follicle, the paracortex, etc. We have already talked about it. But the mature form of the T cell is known as plasma cell. So just like you don't look similar when you are right now or when you just pass the school, you look different, right? So the B cells also look different. So B cells get more mature and differentiated to become plasma cells. By differentiated, I mean that they are more directed to a particular fate, okay? So these plasma cells are actually antibody producing factory. They really produce a hell lot of antibody and secrete them, okay? Now we talk about the natural killer cells. They generally are the lethal weapon of the immune system, because what they do is kill. They are born to kill. Their default fate is killing. They engage to anybody and try to kill unless and until that particular cell says, okay, I have specific region, I have specific reason not to kill me because I'm your body's cell, I'm your self cell. Until and unless that reason is specified, they would engage to anybody and kill them. And that is why they are having very good properties in terms of tumor immunity. So tumor could be also attacked by the NK cells. And as we go along, we would also learn about NK cells in this lecture, okay? Just to give you a premiere that how NK cell and the CD8 positive cytotoxic T cells are different, because both of them are actually cold blood killer, right? So how they are different. So the CD8 positive T cells detect that something has gone wrong based on some antigens which are displayed on class 1 MHC. Sometimes what happens is like the cells which are infected by some virus or other pathogens that ultimately hijack this MHC production pathway and don't allow this cell to produce class 1 MHC. As a result, what happens? Class 1 MHC is not displayed on the surface. Now, CD8 positive cell would always scan for class 1 MHC is there or not. If class 1 MHC is there and they see a viral peptide or any pathogenic peptide is displayed on class 1 MHC, that means, okay, something has been wrong and I should engage here and kill that. But if the MHC is downregulated, they would think, okay, everything is fine, there is no attack. But here is the fail-safe mechanism, which is natural killer cell. Natural killer cell are clever. They know that every cell of our body should express class 1 MHC molecule. Any epithelial cell, any liver cell, or etc. can express the class 1 MHC. So as a result, what happens? They sort of attacks the cell which don't have class 1 MHC. So if our CD8 positive killer T cell fails, NK cells save the day. This is how beautifully our immune system is like wired, such that it can really maximize its response against pathogen and save our body from several pathogens and harmful attack. Let's talk about these cells, all of the derivatives of myeloid progenitors. Eosinophil, mast cells, neutrophil, basophil, all of them are involved in somewhat to the innate immune system. Among them, specifically, mast cell is very important in context of inflammatory response and allergic response. And they did ask question on which of these following cells is associated with type 1 hypersensitivity or allergy. So this is an important concept to know, that these are important in context of inflammation, modulation of adaptive immune response, and allergic response, the type 1 hypersensitivity response. Especially mast cells has receptors which are known as IgE receptors. IgE antibodies are especially important for all sort of allergic response. Okay, not only mast cells, basophils has the same receptor, so they can play similar role in terms of in context of the in context of allergy. Okay, so what they do is they secrete a lot of granular substance known as basophil, known as histamines. So I'm a bit mumbling today because it's my first class in YouTube, the live class, so I don't know, but as I go along I think it would be more clear. Next, we talk about the communication between all the divisions of the armies. So we know we have land troopers, we have navy, we have air forces, but all of these need to connect to each other, right? And they talk to each other by walkie-talkie or radio communication. But have you ever wondered that how these immune cells talk to each other? Like, for example, how a immune cell, which is a component of your adaptive immune system, talk to your innate immune system cell? What is the intercom for them? The intercom or the walkie-talkie for them is actually cytokines. So the cytokines plays very important role in terms of communication between several immune system. Okay, so the communication strategy that they take is cytokine mediated responses. So that pretty much summarize about the immune system, like the cells and the organs of the immune system. Now what we should start is try to understand the development of the T cell and the B cell. So we would go really slowly because it's a very long topic, so we'll go slowly. So now we are going to talk about T cell and B cell, and before that we just refresh our memory that we know from the bone marrow stromatal, so from the bone marrow endosteal niche, the immune cells are developed, right? Inside the bone marrow there are two locations, something associated with the osteoblast and something associated with the blood vessels. Now, things which are associated with the osteoblast have the hematopoietic pluripotent stem cells. Hematopoietic pluripotent stem cells actually give rise to the immune system. Now, hematopoietic pluripotent stem cell give rise to two important progenitors, lymphoid and myeloid. This we have already learned because they have asked several questions from this particular topic in IIT JAM, JNU, and many other examinations. That okay, which of the following cells are derived from lymphoid origin or myeloid origin? Then you have to understand, and you have to keep this particular slide to associate with your memory, right? So Here, the lymphoid progenitor gives rise to broadly the cells of your adaptive immune system, such as your T cell, B cell, etc., NK cells. Now, myeloid progenitors give rise to the cells of your blood, such as RBC, your platelets, and also other components of your innate immune system, such as your mast cells, basophils, other granulocytes as neutrophils, etc. So we already looked at it, right? Now, from lymphoid progenitor, T cell, B cell precursors are born, and from that they get secreted to the bloodstream, and they go to other organs such as thymus, where T cell development can take place, right? So in this video we have a focus on how T cell development take place first, and then we are going to talk about how B cell development took place. Now, B cell precursors are getting secreted as well in an immature state. Now, B cells are less immature than the T cells at a comparison. So T cells would be really immature stage when they leave the bone marrow, because their most of the development takes place in the thymus. But B cells, some portion of their development has already taken place in the bone marrow, and now they have expressed the minimum requirement to be a B cell, and they would be now secreted. Okay, so from there, where they go? They actually go to the nearby lymph node. Now, lymph node are actually present all of our body. When the T cell and the B cell are in the circulation, they go to the lymph node. Okay. Now, however, there is another cell type which is also formed from the lymphoid progenitor, which is NK cells, right? Now, myeloid progenitor, as I remind you that they give rise to myoblasts. From there, eosinophil, basophil, neutrophil, etc. are produced, right, in sequential step. We are not going to talk about the other derivatives such as like the blood cells, because blood cells means the RBCs are not our point of discussion today. But anyway, myoblast can also give rise to monocytes, which can become macrophages. It can also give rise to megakaryoblast, which ultimately secretes the platelets. Okay. Now, erythropoiesis process also takes place from myeloid progenitor cells. It's amazing that same myeloid progenitor can differentiate into different lineages, and how this lineage is committed, how this is formed, this is not the point of our discussion, because this is not in the syllabus. So we focus strictly on what is in our syllabus, and we discuss that. Fine. So one important thing to know, that both myeloid progenitor and lymphoid progenitor can give rise to one of your innate immune system component, which is your dendritic cell. Okay, so dendritic cell is one of your innate immune system component. Dendritic cell is an antigen presenting cell, and it's named dendritic cell because it has an appearance of dendrite like projection that you see in neurons. So that's why they are known as dendritic cells, but they don't have dendrites or axons like the neuron have, right? Okay, so now let's focus on T cell development. So the T cell development starts in the bone marrow, because the T cells are born in the bone marrow. From where? From the lymphoid progenitors, which are actually derived from hematopoietic pluripotent stem cells. Now, the T cell development would take place more or less in the thymus, right? So T cells are like specialized armies. I mean, they have to be either these SWATs, which are like the cold blood killers, the cytotoxic T cells, or they would be the helper T cell, the reserve branch of the army, right? But T cell at this particular point of time doesn't know what it would become, so it's like still at very immature state. So it has to go to its training school. So let's send this T cell to its training school. Okay, so now our immature T cells, we call them T cell precursor. So these terminologies are a bit confusing. For simplicity, we're going to tell them that they are T cell precursor, and they are not actually mature T cell yet. So there are a few terms that we are going to talk about: a mature T cells, a naive T cells, a T cell precursor. T cell precursor doesn't have all the characteristic of a T cell as well. A naive T cell has all the characteristics. It has a functional receptor, but it is still inexperienced, that it don't know how to interact with the antigen presenting cells or activate them, or how to activate a B cell, so they don't have that experience. And then comes the mature T cell, which has both the combat experience. Okay, so it's very similar to the army divisions, armies which are like have all the skills but never applied their skills. And so the naive T cells are like this. So these concepts would like reappear in these videos and today's continuous session for multiple time. Okay, so now we have our thymus. Inside the thymus, if we do a cross section, we would look, the thymus would look something like this. Let's look at the anatomical region for a second, because it is so important to understand the anatomy to better understand how the T cell development takes place inside. So this is how a portion of the thymic cross section look like. You have different anatomical regions. Starting from the outside, there is subcapsular region, there is a cortex, there is a corticomedullary junction, which is a transient zone, and there is medulla. So where does T cells come from? So there are many afferent and efferent blood vessels into the thymus, which brings in these T cells and which are actually secreted from the bone marrow. Now, from hyoendothelial venule, T cells come in, but once they come in, they don't go from outside, so from inside to out manner. They first go to the outside and get accumulated in the peripheral region. There they hold their breath and stay there. So they are populated in the subcapsular region. From that, they eventually come down across the cortex, then corticomedullary junction, then to the medulla. So in this all transition from subcapsular region to cortex, cortex to corticomedullary junction, each of the cases, they are interacting with some sort of thymic epithelial cells. And these thymic epithelial cells are working like a teacher. Okay, so the thymic epithelial cells teach them many things. So let's see what the thymic epithelial cell teach them and how they really select that. So if it's a school, there is a pass-fail system, there is a selection category, right? So let's try to understand. Okay, so at least this thymic training takes 3 weeks. So it's not easy, right? So the T cell maturation process is not so quick. It's pretty time consuming. So first, we are at the subcapsular region, which is the peripheral most layer of your thymus. In the subcapsular region, you have your T cells. At this particular cell, they're very much immature. They are known as double negative T cells. Why they are double negative? Because neither they have the CD4 positive receptor, co-receptor, or they don't also have CD8 positive co-receptor. Either or, they don't have CD8, don't have CD4, so they have nothing, right? They only have the T cell receptor, but the T cell receptor is not ready yet for recognizing things. That's why they are known as double negative. Okay, at this particular stage, TCR is present. Okay, there could be alpha beta type of TCR or gamma delta. So gamma delta is a complete different type of T cells, which we don't want to talk in this video. Now there are also different stages of these T cell development, such as DN1 stage, 2, 3, and 4. But how they are different from each other, all these stages. So as the T cell or the very mature T cell progress along its growth phase, what happens is these double negative stages are also changing. Their marker expression, the surface marker expression, are changing. So based on what type of surface molecules are expressed in these cell types, they are categorized differently. But what is the important thing which you need to know for IIT JAM, that at this particular point, at the double negative stage, the VDJ recombination takes place for TCR gene. Wait a second, what are you saying? VDJ recombination for T cell receptors? Yes, you heard me correct. So there is a question in some year that when we review the questions, you would get to know that they talked about VDJ recombination takes place in the antibody genes, then the B cell receptor, then there was also option of T cell receptor. So everybody happily ticked and it was a MSQ answer. So everybody happily ticked all those B cell related stuff or antibody genes or B cell receptor. Nobody cared that also T cell receptors undergo the same VDJ rearrangement. Why do you think they need to undergo VDJ recombination? So as we go along this class, as we learn more about how T cell is recognizing the pathogen derived antigen from MHC, we would appreciate why VDJ recombination is important for T cell as well. And this concept is not well taught in a school or colleges, so we'll clear that concept. So for now, we try to understand that what is the sequence of VDJ recombination in the T cell. So the beta chain of the TCR gets rearranged, then the alpha chain in a sequential fashion, and what happens, they combine and form the mature TCR alpha and beta receptor. However, the recombination possibility of gamma delta is so low, so that's why we have a very small cohort which is known as gamma delta T cell, but majority of them having these TCR alpha beta. Now this TCR alpha beta has to recognize MHCs, because that's the important thing. They have to understand who is bad, who is good, right? Who is self versus who is non-self. Anyway, so the TCR gamma delta cells are very low in number. And why they are low in number? Because it has an explanation, but I'm not going to give it to you in this particular class, because I think for IIT JAM this concept is not so important to understand why gamma delta T cell are so low in number. But anyway, we were stuck in subcapsular region for a long time. Now we are going to move on from subcapsular region to a double positive stage. Now why they are double positive? Double positive because they have both the receptor, both the CD8 and CD4 co-receptor. Both receptors are there. Okay, so that is why they are double positive. Now double positive T cells residing in the cortex. So they have descend down from subcapsular region, which is the peripheral most region, to the cortex. Now inside the cortex, they have specific cells called thymic epithelial cells, right? These thymic epithelial cells help these double positive cells and teach them an important lesson. In turn, they take the exams as well, and either some of them fails and pass. A point to be noted that most of them actually fail and dies. 80% of the cells of the thymus are actually these double positive T cells, suggesting this double positive state or double positive stage is so important. So let's see why they are so important. Let's see how they interact with the thymic epithelial cells. So this is the enlarged view of the thymic epithelial cells. Now this thymic epithelial cell has both class 1 MHC and class 2 MHC. Now first, there are two distinct selection processes going on. First, there is a positive selection where the cells learn to recognize MHC molecule bind peptides. They first need to learn that, okay, how to understand this is a gun, right? Army need to understand how a gun look like, right, in order to fire a gun. So here the basic criteria is like you at least have to recognize a MHC. Doesn't matter it has a self molecule or a pathogenic molecule on displayed on its surface, but you have to understand what is a MHC. Now you can only recognize a MHC with a moderate affinity. If your affinity is too high, you are dead. If your affinity is too low, you're also dead. So these are the two selection processes, positive and negative selection. Let's look at the positive selection again. In this phase, they learn to recognize MHC molecule, otherwise they would be neglected. And the next round of selection won't be there for the cells which cannot recognize. Only the cells which recognize some MHC bound peptides would go to the next round of selection, and they would like really expand clonally. So let's see, I mean, what is the next step. Now the cells which are not recognizing these things, which are majority of the cell population, they would undergo death by neglect. So 90% of the cells of these DN positive stage, sorry, DP double positive stage, they die because they cannot recognize MHC. Now if an army cannot fire a gun, then how he is an army? and how he's going to work in the battlefield, right? So they are not qualified in the test, so they have to be dead. So immune system is like even more stringent than our defense system, right? Okay, so now we're going to talk about how these cells are going to interact with the thymic epithelial cells. One possibility is they interact with a low affinity, low to moderate affinity. Then they would go to next round of selection, which is the negative selection. But if they interact with a very high interaction, they would die. Okay, the next round is happening in the medulla. Inside the medulla, the cells would have both class 1 and class 2 MHC. If a double positive cell try to recognize peptides present in class 2 MHC, then what happens is like these cells would stop, I mean, expressing CD8 co-receptor and start expressing or upregulating CD4 expression. Okay, and it becomes a CD4 positive T helper cell. Now the other way around, if it recognizes class 1 MHC bound peptide with a moderate affinity, and they would start expressing the receptor which is CD8. It is CD8 gets upregulated and CD4 gets downregulated. Now that is how they're selected. Either they become a CD helper cell, a T helper cell, which is expressing CD4 positive markers, and otherwise they become a killer T cell, which is expressing a CD8, right? Now there are possibilities which interacts both of them. That's a problem, and they would die by neglect, right? They can either recognize both of that, both of class 1 and class 2 MHC bound peptides, or they can recognize none of them, and both of these situations the T cells won't survive, they would die. So it turns out if you begin with a number of 100 immature T cells, only 1% to 2% survive and go to the next round and eventually get posted to the peripheral lymph organ, which is your lymph node, right? Okay, so we again look at it. High affinity interactions, they are dead. Only the moderate affinity interactions are alive. So eventually they would move using the bloodstream and get posted to the peripheral lymph organ, which is also a secondary lymph organ, which is known as the lymph node, which is localized all of our body. So all of our body is like populated with several small army checkposts or checkpoints, or let's call them army base, small army base. Okay, anyway, so here we're going to talk about T cell maturation process. So we so far talked about T cells, how they develop inside the thymus, but eventually we learned that after their training is done in the thymus, they're moving out to where? Moving out to be posted in the secondary lymphoid organ, such as our lymph node. Now let's see how pathogen and cytokines can really help in the process of T cell differentiation and maturation. So here there are two cytokines, a red one and the blue one. Both these cytokines can really have opposing effect in terms of what type of T cell it would become, right? Depending upon the particular cytokine it is interacting with, the T cell can become two different fate. It can have two different fate. Even if they are helper T cell, they can have two subgroups. Let's imagine, and it is totally pathogen dependent. Let's imagine our pathogen is a virus in this case, a virus which has double-stranded RNA. Okay, so this viral RNA would be actually detected by the TLRs, which are the toll-like receptors. Now they have to actually get detected. TLR signaling take place inside the nucleus. There are specific factors which are produced. One such factor is interleukin 12. Interleukin 12 is secreted and binds to interleukin 12 receptor. Now T cell receptor activation takes three important signals, okay? So first of all, they require a TCR versus MHC interaction, MHC class 2 interaction. Second, they require a CD80 and CD80 ligand interaction, CD28. So these gives two important cell signals, and the third signal comes from actually the cytokine receptor and cytokine mediated signaling. So all these three signals combinedly is conveyed into the nucleus, and that actually transcribe a master regulator. This master regulator would specify the sublineage of all these T cells. For example, in this particular case, this T-bed master regulator would force the T cell to become a Th1 cell. Okay. Next, we talk about another type of pathogen. Here, the pathogen is a worm, a helminth. Okay. So now they're also getting recognized by other TLRs. Other TLR signaling leads to actually secretion of other cytokines such as our IL-4. IL-4 is very different from IL-12, so IL-4 has different receptor, IL-4 receptor. Now both the signal 1 and signal 2 was more or less similar, but the signal 3 is different, and as a combination of both, other gene is transcribed, other master regulator, which is GATA3, and which push the fate towards a Th2 helper cell. Okay, so we at least learn the take-home message is, depending upon what type of cytokines are secreted and what type of cytokines are experienced by this T cell, they are going to differentiate into different fates. Okay, either Th1 fate or Th2 fate, T regulatory fate or Th17 fate, and all have different functionality, but they are all CD4 positive T cell, a naive CD4 positive T cell. Naive CD4 positive T cells are inexperienced, but they are molecularly they are pretty much done. They know how to recognize MHC derived peptides, they know how to interact with B cells, so they know stuff, but they haven't done it in the battlefield, so they are inexperienced, but still they can get specialized into specific cohorts. Okay, now what happens really at the molecular level is one cytokine signaling is repressing the alternative fate. For example, here if you think about the T-beta, which is produced by the STAT4 signaling, you see the T-beta master regulator is actually blocking IL-4 mediated responses, IL-5 mediated responses, but enforcing interferon gamma mediated responses. Whereas the opposite thing happens for another master regulator such as GATA3. So when GATA3 is expressed, it ensures more IL-4 is there such that more autocrine signaling can take place. Other signaling which would differentiate to a Th2 lineage, they are preferred, but the signaling which would actually give rise to a Th1 fate, they are suppressed. So this is why there is a mutual opposition between the two transcription factors that are transcribing these things. So that is why they have to choose their lineage. I would be differentiated to in this particular lineage or this particular subgroup. That is how the T cells actually decide. Okay, so that pretty much summarize our talk about T cell development. So it's a big topic, T cell development, but we really covered it into a span of half an hour. T cell development, T cell activation and maturation. We also got an introduction about the immune system cells, cell types and organ types. So in next lecture, we're going to focus on the other aspects. So for that, I would like to take your leave and see you in the next lecture, which is from 9:30. So bye everyone.